Hi everyone, welcome back to this educational channel called Learn Bio with Janet. Today we'll be discussing another type of nuclear division other than mitosis. This nuclear division is called meiosis, and it is a type of nuclear division that results in the formation of gametes such as the egg cell, which is haploid, and the sperm cell, which is also haploid. So we are going to study the definition of meiosis, where it occurs, and its importance in this first video. And in the second video, we will study the process of meiosis. This topic is from the Form 4 KSSM Biology syllabus, and the chapter involved is Chapter 6, entitled Cell Division. The learning outcomes for this lesson are as follows. After this lesson, you should be able to state the meaning of meiosis. Number 2, be able to identify types of cells that undergo meiosis. Number 3, be able to state the necessity of meiosis in A, the formation of gametes, in the process of gametogenesis. B, the necessity of meiosis in producing genetic variation. And C, the necessity of meiosis in maintaining the diploid chromosomal number from one generation to another. What is the definition for meiosis? Meiosis is a type of nuclear division that occurs in reproductive organs to produce gametes, which are reproductive cells. And these gametes or reproductive cells contain half the number of chromosomes of the parent cells. So the gametes are said to be in haploid condition, as they have only half the number of chromosomes as that of the parent cells, which are said to be in diploid condition. Thus, in meiosis, one diploid parent cell produces 4 haploid daughter cells, which will develop into gametes. Examples of gametes are sperm, ovum, and pollen. So here we have a diploid parent cell, and it undergoes meiosis. Meiosis consists of meiosis 1. The first cell division is called meiosis 1, and the second cell division is meiosis 2. And thus, 4 haploid daughter cells are produced at the end of meiosis, which are genetically different from the parent cell. So how is it genetically different and why? We will discuss in the second video. Let us discuss a few basic differences between the two types of nuclear divisions, which are mitosis and meiosis. In mitosis, a diploid parent cell undergoes one nuclear division, and this is followed by a cytoplasmic division, so that one cell divides into 2 daughter cells that are genetically identical to the parent cell. And the daughter cells are also diploid, like the parent cell. They are the normal body cells or the somatic cells, such as the skin cells. In meiosis, a diploid parent cell undergoes 2 nuclear divisions, that is, meiosis 1 and meiosis 2. So after every nuclear division, like meiosis 1, there is a cytoplasmic division. After meiosis 2, the nuclear division called meiosis 2, there is another cytoplasmic division, where the cell divides into 2 again. Thus, 4 haploid daughter cells are formed at the end of meiosis, and they are genetically different from the parent cell because of processes that occur, such as crossing over, which we will study in the next video. Thus, if we were to compare the two processes, the first difference is that mitosis produces only 2 daughter cells, but meiosis produces 4 daughter cells. In mitosis, the daughter cells are diploid. In meiosis, the daughter cells formed are haploid, only having half the number of chromosomes compared to the parent cell. In mitosis, the daughter cells are genetically identical to the parent cell, but in meiosis, the daughter cells are genetically different from the parent cells. We will find out how this occurs in the next video. Let us briefly discuss what happens in interphase and in the process of meiosis. So this is a brief explanation on meiosis, and that is what happens in meiosis 1 and 2. If you are not very sure about the process, do not worry, as there will be a second video coming up on the more detailed explanation about the process of meiosis. Okay, so let's take an example of a cell with 2 chromosomes. These 2 chromosomes have the same length, the same shape, and the same position of centromere. They carry the same type of genes. So we call these 2 chromosomes a pair of homologous chromosomes. Homo meaning the same. Now, one copy comes from the father or the male parent. It's called the paternal chromosome, and one copy of these chromosomes come from the mother or the female parent, and it's called the maternal chromosome. Okay, so in this cell there are 2 chromosomes, and this is a diploid cell as there are 2 copies of this type of chromosome. So we say that 2n, the diploid number, equals to 2 for this cell. 2n equals to 2. Now during interphase consists of 3 phases, G1, S, and G2 phase. So the first phase is G1 phase, when the cell is still small and young, and the chromosomes are not yet duplicated, so they are unduplicated. And it's just like they are only made up of 1 strand of DNA each. Okay, 1 strand of DNA, which is very long. Alright. But after G1 phase, we have S phase, where there's duplication of chromosomes. That means the cell makes another copy of each chromosome. Or we say there's replication of DNA during the S phase. So here's what happens. Each chromosome now, after duplication, consists of 2 sister chromatids joined at the centromere. So it's still 1 chromosome, 1 whole chromosome. This one X-shaped structure is 1 chromosome. 1 X-shaped structure is 1 chromosome. So 2n still equals to 2. 2n equals to 2, although the mass of DNA has doubled. Let's say you look at the first chromosome here, the mass has doubled because now it's made up of 2 sister chromatids. But the number of chromosomes is still the same. This is 1 chromosome, which is X-shaped like that, made up of 2 sister chromatids, is considered 1 chromosome. Okay, so 1, 2. Now what happens in meiosis 1, the first nuclear division? During the first nuclear division, the homologous chromosomes here will separate from each other and move to different poles. Maybe this black chromosome moves to this pole on the left, and this white one goes to the pole on the right. Okay, when the spindle fibers shorten and contract and pull them apart. So this happens in the phase called anaphase 1. Homologous chromosomes separate and move to opposite poles of the cell. Then cytokinesis occurs. The cell divides into 2. So the result is 2 daughter cells. 2 daughter cells, and in each daughter cell there's only 1 copy now of each chromosome. Okay, 1 paternal chromosome in this cell and 1 maternal chromosome in this cell. Okay, we say there's only 1 copy of each chromosome in each cell. So this is called the haploid condition. Alright, so from diploid condition, now the cell has become haploid in the first nuclear division or meiosis 1. Now after that, there's meiosis 2. In meiosis 2, the sister chromatids separate during anaphase 2. Okay, for example, this sister chromatid will move will be put to this pole here, and the other sister chromatid is put to the other pole when the centromere divides. At first, in meiosis 1, centromere does not divide yet, but in meiosis 2, centromere divides. Then the sister chromatids are pulled apart and separated and moved to opposite poles. So the end result is that for each cell here, 2 daughter cells are produced. For this cell, after cytokinesis, 2 daughter cells are produced. Okay? So after meiosis 1, there's cytokinesis. Then after meiosis 2, there is cytokinesis. After meiosis 1, there's one cytokinesis. And after meiosis 2, there is again cytokinesis. So the end result is that each chromatid becomes a new chromosome, and the daughter cells formed finally also have one chromosome each. So they are haploid cells. There are no 2 copies of the same type of chromosome. There's only one chromosome in each cell. So it's called a haploid cell, and n equals to 1. Okay, that means in this haploid cell, there's only one chromosome. So to round up, in meiosis 1, homologous chromosomes separate. In meiosis 2, sister chromatids separate. Now we've studied the definition for meiosis, and we have also discussed the basic process that occurs in meiosis. Let's go on to talk about the site or the location of meiosis. So where does meiosis occur? Meiosis occurs in reproductive organs of animals and humans, that is, in the testes of males and the ovaries of females. For plants, it occurs in the flower parts, that is, the anther, as seen here, which is usually covered with a yellow powder, and that yellow powder is the pollen, which contains the male gametes produced through meiosis. Also in the flower, there is an ovary, which may be in the same flower or it may be in a different flower from the flower that has the male parts. But anyway, for the ovary or the female structure, inside it there is an ovule, and inside the ovule, the ovum is produced through meiosis also. Alright, so ovule in ovary produces the ovum. This is the location of meiosis. The ovum is produced through meiosis inside the ovule, which is inside this structure called the ovary. We have already discussed the definition and the location of meiosis. Now let's ask this question. What is the importance or the role or the need for meiosis in organisms that carry out sexual reproduction? So in sexual reproduction, there must be a male gamete, for example, a sperm, that fertilizes the female gamete or ovum. And when there's fusion of the 2 nuclei, a zygote is produced, which develops into the offspring of the organism. Okay, so all that is involved in sexual reproduction. So in sexual reproduction, gametes must be produced, and meiosis produces these gametes, which are haploid reproductive cells that are involved in sexual reproduction. And these gametes are produced during the process of gametogenesis. So simply put, gametogenesis is the process of formation of gametes in the testes and in the ovary. Okay, so this overall process of formation of gametes involves meiosis as one of its main processes. We will study more about gametogenesis. We'll study more of gametogenesis in another chapter. Okay, right. So meiosis produces gametes or haploid reproductive cells, such as the sperm and the ovum for animals and humans, and for plants is the pollen and the ovum. These are all gametes. The pollen, inside the pollen there is a male gamete for plants. Right. Meiosis ensures that the diploid chromosomal number of an organism that carries out sexual reproduction is maintained from one generation to the next. This is the second role or function. So we will be discussing more of this in the next slide. But just briefly, it means that when the sperm, which is haploid, fuses with the ovum that is also haploid, a zygote is produced that is diploid. So sperm has one set of chromosomes, ovum has one set of chromosomes. Okay, sperm has the set of chromosomes from the father, so it has the paternal chromosomes. Ovum has the set of chromosomes from the mother, which are the maternal chromosomes. Okay, so when they fuse together, the zygote will have two sets of chromosomes. One from the sperm or the father, and one from the ovum or the mother. So the zygote now becomes a diploid organism. Okay, so meiosis ensures that the diploid chromosomal number of an organism is maintained from generation to the next because it produces haploid sperm and haploid ovum. So that when these two haploid cells fuse together, you get the diploid zygote and a diploid organism formed. Now we'll discuss more of this in the next slide. Now number 3, meiosis produces genetic variation among organisms of the same species through crossing over in prophase 1 and random arrangement of homologous chromosomes in metaphase 1. The word variation means differences between individuals of the same species. For example, differences between humans. So let's say we have a family where there are 2 daughters. So here we can see that, for example, the children, although the children of a family that comes from the same parents may have different features from each other. So one may have brown hair, the other may have black hair. One may have straight hair, the other curly hair. And one may have dimples on the cheek, but the other may not have. And even the face shape may be different. So why are there so many differences between individuals of the same species and even between those children that come from the same parents? For example, you can compare yourself with your siblings or your brothers and sisters, and you'll be able to spot many differences between you and your siblings, unless you are comparing yourself with a twin, an identical twin. Then identical twins have the same DNA, and there is no variation for identical twins. All right, or very little variation. Okay. Now, but if we are talking about siblings in the family that are not identical twins, we can see that there are many different characteristics. Even the blood group may be different. So why is that so? Because in meiosis, when the gametes are produced, for example, when the sperms are produced and the ovum are produced in the parents, each ovum that's produced in the mother is different from another ovum, is genetically different from another ovum. Each sperm produced in the father is genetically different from another sperm in the father. So this is because meiosis is involved to produce genetic variation, okay, through the process called crossing over, which we will study in the next video, and through random arrangement of homologous chromosomes in metaphase 1, okay, which we will study in the next video when we talk about the process of meiosis. Let us try and answer this question. How does meiosis maintain the diploid chromosomal number of an organism in the zygote formed after fertilization? And how does meiosis maintain the diploid chromosomal number of an organism from generation to generation? So firstly, we must be aware that all normal individuals from the same species must have the same diploid chromosomal number. Sometimes it's just called same chromosomal number. which must be maintained from one generation to the next. That means the diploid chromosomal number must always be the same from one generation to the next. It must be maintained. For example, we have already studied in the previous videos about different organisms which have different diploid chromosomal numbers, which are fixed and maintained from generation to generation. For example, siamese, which is corn, has 20 chromosomes in each diploid cell. Gallus or chicken has 78 chromosomes in each diploid cell. And Oryza sativa or rice has 24 chromosomes in each diploid cell. Another example is the tiger, Panthera tigris. It has 38 chromosomes in each diploid cell. So the diploid chromosomal number is fixed and maintained from one generation to the next. Now, what happens if the diploid chromosomal number were to change from one generation to the next? So even if there is just one extra chromosome in the cells of an individual compared to a normal individual of the same species, it causes an abnormal situation, and an abnormal individual is produced that may have serious health problems. For example, we know that a human being has the diploid chromosomal number of 46. A normal human being will have 46 chromosomes in each diploid cell, whereas a Down syndrome child has 47 chromosomes. Down syndrome child has an extra chromosome number 21. So the child has 47 chromosomes in each diploid cell. And this child suffers from mental retardation. The child is mentally retarded. And there are also other health problems related to a Down syndrome child. So it is very important for each organism or each species of organism to always maintain the diploid chromosomal number from one generation to the next. For example, from the father or mother to the child, the chromosomal number must be maintained. Let's continue to discuss how meiosis maintains the diploid chromosomal number of an organism in the zygote after fertilization and also from generation to generation. In this picture, we see the human life cycle. So, a diploid cell in the testis of a father undergoes meiosis to produce a sperm cell. In the process, the number of chromosomes is reduced from 46 to 23. The diploid cell has two sets of chromosomes, whereas the sperm has only one set of chromosomes from chromosome number 1 to 23. So the sperm is formed through meiosis, and we call meiosis a type of reduction division, because when the sperm cell is produced through the division of the diploid cell, the number of chromosomes is reduced to half that of the parent cell. Similarly, a diploid cell in the ovary of the mother undergoes meiosis to produce an ovum which has half the number of chromosomes compared to the original diploid cell or parent cell. So once again, the diploid cells in the ovary of the mother will have 46 chromosomes each, or 2 sets of 23 chromosomes. But after meiosis, when the ovum is produced, it only has 23 chromosomes, or 1 set of chromosomes. So the ovum is haploid because it has only 1 set of chromosomes, same like the sperm. Now, why is it that meiosis is involved and there is reduction in the number of chromosomes? So meiosis is important because it produces the haploid gametes with half the number of chromosomes compared to the parent cell. This means that the number of chromosomes have been reduced to half that of the diploid cell in the parent. This helps to ensure that in sexual reproduction, when the sperm and ovum fuses together, the nuclei of the two gametes will fuse. Then the chromosomes from the sperm will combine with the chromosomes from the ovum in one nucleus, the nucleus of the zygote, which is formed through fertilization. So the nucleus of the zygote will have 2 sets of chromosomes. One set is from the sperm of the father, and the other set of chromosomes come from the ovum of the mother. So the zygote is said to be diploid. And we can see that when 23 chromosomes from the sperm combine with another 23 chromosomes from the ovum, the zygote will have 46 chromosomes. So that the diploid number of chromosomes is produced in the zygote and is maintained from the father to the offspring, because the zygote will become the offspring of the father and mother. So the offspring will inherit the traits from both parents through the sperm and ovum, because the zygote or offspring will have both sets of chromosomes. One set from the father, so it will inherit some characteristics of the father, and one set of chromosomes is from the mother, so it will inherit some characteristics from the mother. Then the zygote will undergo mitosis to produce many more cells that are diploid, and this will increase the size of the offspring, and that is growth occurs. So finally a baby is formed, and this baby will grow and continue to undergo mitosis. The cells of the child will undergo mitosis to produce more and more cells for growth, and so the size of the offspring will continue to grow until he or she becomes an adult, through the process of mitosis. Now let's say that in the production of gametes, in this part, let's say that mitosis is used to produce the sperm and the ovum. So if the gametes are produced by mitosis, each gamete will contain the diploid chromosomal number. That means the diploid cell of the father will produce a diploid sperm if it is produced by mitosis. And then 2n will be equals to 46. There will be 46 chromosomes in the sperm, and there will be 46 chromosomes in the ovum. So after fertilization, when the sperm and the ovum fuses, that means the male gamete and the female gamete fuses, the zygote and offspring formed will have twice the chromosomal number of the parent organism, because 46 plus 46 chromosomes will produce 92 chromosomes. Alright, so the zygote will have 92 chromosomes, and this offspring will definitely not be a human, having so many more chromosomes than the normal individual in each cell. Therefore, in order for the offspring or the zygote to possess the same chromosomal number of the parent, 46 chromosomes in each cell. Meiosis is important. The gametes must be formed through meiosis and not mitosis. This is how meiosis helps to maintain the diploid chromosomal number of an organism in the zygote, and from one generation to the next generation, from father to child, and from the child to the child's children, and so forth. Let us discuss another example other than human beings. Let's imagine that there's an organism where the diploid number of chromosomes is 6. It means that there are 3 different types of chromosomes. 2, n equals to 6, n equals to 3. There are 3 different types of chromosomes, but in the diploid cell, there are 2 sets of that chromosomes. However, in the formation of gametes, the chromosomal number is halved. So, from 2n equals to 6, now the gametes formed, such as the ovum and sperm, have only 3 chromosomes, where n equals to 3. Okay, as shown here, the haploid ovum has a haploid condition where n equals to 3. When n equals to 3, it means that there are 3 different types of chromosomes in the cell, and they are all carrying different types of genes, and their lengths are not the same. Okay, you have the long chromosome, the medium chromosome, and the short chromosome in this ovum. Likewise, for the sperm, n equals to 3, so it has also got 3 chromosomes of different lengths and carrying different types of genes. Now, in fertilization, the ovum and the sperm will fuse together, and this will ensure that the zygote form has the diploid number of chromosomes, because the 2 sets of chromosomes will combine together in the nucleus of the zygote, and therefore the zygote is a diploid cell, where 2n equals to 6, as shown here, due to the combination of chromosomes from the sperm, the haploid sperm, and the chromosomes from the haploid ovum. Okay, so we get back the diploid number of chromosomes, which is 2n equals to 6. And this shows that in the formation of gametes, the number of chromosomes is reduced to half, so that after fertilization, when the sperm and ovum fuse together, you get a diploid zygote, where there are 2 sets of chromosomes in the zygote. We have already discussed 2 examples of how meiosis helps to reduce the diploid chromosomal number of the parent cell to half in the formation of the haploid gametes. And this is the aim of meiosis, which is a form of reduction division, meaning that it's a type of nuclear division to reduce the number of chromosomes in the gametes to half of that of the parent cell. Now, in the fusion of the 2 haploid nuclei of sperm and ovum, or gametes, during fertilization, a diploid zygote will be produced, and hence the diploid number of chromosomes is maintained from generation to generation, as the zygote will develop into the offspring of the organisms. Now, in this picture, we see an egg cell with the nucleus, which has the haploid number of chromosomes. Okay, and here we see a sperm that has already entered the egg cell. The nucleus of the sperm has entered the egg cell, and it is also containing the haploid number of chromosomes. So when the nucleus of the sperm fuses with the nucleus of the egg, it will produce a zygote with the diploid number of chromosomes, which is the same as the condition of the parents that are diploid organisms. That's all for this video. In the next lesson, we'll continue to study the subtopic of 6.3 in Chapter 6, entitled Cell Division, for the Form 4 KSSM syllabus. We'll be discussing meiosis part 2, and that is the process of meiosis. So thanks for watching. Stay tuned for the next video. Do share, like, and subscribe. See you soon.